So it's my pleasure to introduce today Dr. Aaron Ragsdale, who joins us from the University of Wisconsin. I met Aaron when he was a postdoc with Simone Gravel at McGill University, and he focuses a lot on methods development, specifically for demographic inference and thinking about both demographic inference and selection in its various forms, background selection, stabilizing selection. I'm trying to convince him to think about balancing selection, but it's a hard sell. And he has contributed to a variety of probably pretty frequently used software. So if you have worked with the Dean's package, you've worked with TS-Kit, MS Prime, all of these wonderful features that are associated with population genetic software. Aaron has been an active user and contributor for a long time, including Moments and Moments LD. So are you going to talk about that today a little bit? A little bit. Okay, great. And yeah, we're just delighted to have him here. And I'll mention that his visit is sponsored by the Letters and Sciences Dean. We just want to thank the Dean's office for helping this visit. Thank you.
Thanks, Brennan. Thanks for inviting me out, tagging me on to the end of PropGen. I saw some of you this last week over in Berkeley at PropGen, so thanks for inviting me out. It's been nice to chat with a few of you here as well. So I'm going to be talking a little bit about demographic inference, but in the first half, and then specifically thinking about inference of population structure in the deep past, and then thinking in the second half a little bit about some of the we layer some models of selection on top of that, specifically stabilizing selection. Can we predict things about the shared architecture of polygenic traits under stabilizing selection and what that might mean for selection on functional alleles and some other things like that. That's a lot more recent, and some of it is kind of work in progress. So I'd be also interested in you telling me if I'm way off base on some of it as well. So, but we'll see. Okay, so I wanted to start with. Yeah, so give you in 2 slides a history of the field of inference of population structure in human origins, which is kind of tough, but a long time before genetic data was available, a lot of the debates and arguments were based on things like it's like the archaeological record, or things like morphological variation that you see in fossils, and then also thinking about not just how population structure was shaped or present in our own species, but how it was related to other related hominid species as well. And so a lot of the early models kind of would take kind of these verbal or conceptual models, and you plot things like this, where you have tubes and arrows, tubes representing populations and arrows that maybe represent migration between populations, and you're testing things like, do we have a recent divergence of human lineages that replace other hominins, or do we see deep structure among our ancestral lineages with or without gene flow? So these are kind of some of the verbal models that they get proposed and then argued about. These can get really complicated. So you might have these really complex lattices that, and you try to relate these to maybe relevant things that you see in the archaeological record or the fossil record or the climate record. And around the 70s, 80s, and 90s, genetic data started coming available to try to resolve some of these long-standing debates. And so you have first the generation of mitochondrial data to some of these build big trees of uniparental markers and ask which of our verbal models seem to be the best representation of what we see in the genetic data. And then fast forward a little bit more, and you start seeing nuclear genomes nuclear data coming available, and being able to at least restrict some of the models that we think are either supported by the genetic data. So we now think, as of the early 2000s, that we can trace most of our ancestry back to a common ancestry that emerged relatively recently or migrated out of Africa relatively recently compared to some of the other models. And so since then, there's been a lot of effort to kind of refine these types of models. So some of these maybe big questions that motivate me and a lot of other people are just asking questions like how and where were past populations structured? Were the ancestral lineage, how much were ancestral lineages related to each other? Which representative fossils in the past are related to some of those ancestral lineages, or are they not related to ancestral lineages? And then maybe more very general questions about how our demographic history, the genetic history, how genobiology, and also how natural selection all interact to shape variation that we see in populations living today. So this might involve understanding divergences and then recontacts or reticulations in the network or tree relating hominid lineages, or distinguishing between different hypotheses of early structure in our species. So we have a lot of different tools to look at genetic data and try to answer these questions. And so some of these are based on, so a very popular approach is based on the site frequency spectrum that looks at single locus variation and kind of discards information about haplotype structure or linkage disequilibrium across the genome. So some of you have probably used some of these methods like DADDY or FAST-SYM-COL. These are like site frequency spectrum-based methods to learn the parameters or infer parameters in proposed models. And so these will be things like size changes, split times, migration rates, and so on. So you propose a model and then parameterize it in some way, and then try to learn the actual values of those parameterized models in this setting. Or you can have different approaches that also use the same type of data, but fit size changes over time, and then try to fit times maybe when the lineages look like they come back together. So that's one approach. There are many other approaches. Another popular approach is reconstructing coalescence times along the genome, looking at the distribution of coalescence times in something like PSMC or MSMC, or maybe for very recent history, looking at the distribution of ancestry tracks and how those are shared among it between individuals. And so these are methods that are all complementary. I think they give you information from different aspects of our history, or different time periods. So looking at ancestry tracks is very relevant to recent time in admixed populations, whereas something like PSMC is going to be more relevant for deeper in the past. One of the approaches that I've been thinking about quite a bit is looking at patterns of two locus statistics, or you might call this linkage disequilibrium. And so when I say linkage disequilibrium, I'm thinking of the collection of statistics that look at pairs of loci with variation and asking about their correlations between these. And so it's capturing something about haplotype structure, but projected down to just pairs of alleles. And so there are a lot of ways to represent this type of data. So this is a figure from Caballero et al. from their software that's called GONE. Some of you might have used this. This is a funny way of representing it, maybe, but this is the inverse of the recombination rate. And so out here, this would be at very loosely linked sites to sites that are much more tightly linked. And what they're plotting is observed data, probably simulated data, and predictions for how one measure of LD changes as you look at different recombination distances. And so from this, you can see that different population histories, bottleneck population or constant population size or expansion, leave their signature in the distribution of LD at different recombination distances. So in this sense, we're using something like recombination rates to give us something like, you think of mutation rates and mutation rates are rising and or mutations are rising at a given rate and being observed in a sample, gives you some information from allele frequencies. And these correlations give you kind of not quite an orthogonal, but a somewhat independent view of similar processes and how they shape haplotype structure in the sample. And so from this, you can look at try to fit these, if you can make a prediction for what these LD decay curves look like under different scenarios, then you can fit parameterized models or perhaps non-parametric models to the size history. And so we've used, so this is where going back to when I was a postdoc with Simon Gravel, where we were very interested in trying to extend these models from a single population setting to a multi-population setting. So we know that different statistics, different LD statistics like D squared or R squared are going to be sensitive to size changes. So I think in the last slide I was showing how D squared was sensitive to size changes. Other statistics we found, these would be what you might call non-canonical LD statistics or non-canonical two-locus statistics, or just different ways of combining haplotype, kind of two-locus haplotype statistics with allele frequencies. You could think of this as a weighted LD statistic, weighted by some polynomial that are the allele frequencies of the two loci. This different statistic gives you kind of a different view of how past population structure shapes haplotype formation. So if you have a model of population divergence with recent admixture, it's not going to show up very much when you look at D squared, but you can see a large effect when you look at these other two locus statistics. And so this is a statistic that we kind of keyed in on, and we're wondering if we could use this to learn about past population structure. This is not a new statistic. I'll just point out that this is already a statistic that has existed in the literature. It was introduced by Hill and Robertson just to be able to compute expectations for D squared, which is the more natural statistic that we look at with LD. And you can solve for it by, if you plug in your population sizes for combination, so they're using slightly different notation, but population sizes for combination rates, mutation rates, you get expectations from the statistic to compare to data. So we found that we were able to take this classical model from Hill and Robertson and extend it to a multi-population setting. You can account for migration between populations after they split. You can account for admixture events to compute these statistics within and between populations in complex multi-population settings. So we get a large collection of these two locus statistics. We get what we've called DZ. We get other statistics that are related to joint heterozygosity at two loci. So you get this large collection of statistics, just like you have potentially a large collection of allele frequency statistics to be able to compute predictions for it from a model and then compare it to data. The benefit of this is these are low order statistics, meaning you don't need very many individuals to estimate these. And you can, because of that, you can kind of go wide and look at many different populations and LD within those populations that cause them. So it's fast to compute and it scales really well. To just give some intuition about what signal we think we're picking up on with this, if we look at something like this where we're sampling a population with some lineages and one or some of them are introduced through an introgression event that looks maybe something like this. We can imagine that the mutation process can sprinkle mutations on these trees. And then if we think about the haplotype structure that comes out of this type of process, we would see something that looks like this. We have a couple samples that have their lineage tracing through this diverged lineage that contributed this haplotype through introgression, we would see a bunch of mutations in strong LD, but kept at low frequency if this was a small introgression event. And so we see something like this, and this is exactly what this statistic picks up. We're picking up on linkage disequilibrium between low frequency variants. So if you've looked at something like the S-star statistic, if that's familiar to you, this is the exact same signal, but kind of collapsed down into a two-locus framework. So we asked if we could use this. We kind of did what everybody's done in the last 15 years and revisit the out of Africa model. So you could ask, does our classic model of population expansion split from a population bottleneck for the out of Africa population and a split with expansion, can we fit these LD statistics with this model? We found, maybe unsurprisingly, that for some of the statistics like D squared, it does a very good job. For other statistics, we were just not able to fit, like this DZ statistic, we were just not able to fit these statistics with this proposed model. So we tried a number of things, and we found that the best way to fit this statistic is including a branch, perhaps that you might call the putative Neanderthal branch that contributes some ancestry to out of Africa populations, and then another branch that also has is indicative of population structure, contributing to, in this case, the West African Yoruba. And by including these diverse branches with gene flow, we get a very good fit to this statistic that is not quite designed, but very, very amenable to learning about population structure. Yeah. So from this, we realize that this is indicative of potentially early population structure in a lineage that is independent of the putative introgression. Yeah, so we turned to seeing if we could. This was kind of a simple application, and we took this and wanted to ask: Can we actually? We know that we could include additional populations. We have the computational ability to do so. So we asked if we could learn something about early population structure within our species history, and this is not something that is all that new. Going back to the mid-2000s, there researchers, especially Jeff Wall and some of his colleagues at the time, who were looking at genetic data that was becoming available, and seeing that there were hints of haplotype structures that were hinting at early population structure or introgression in many different populations. So not just, so both within Europe, putatively Neanderthals, and also within Africa. And they were using the statistic S-star that's picking up on this same signal. More recently, this has been I would say, bolstered by more modern techniques, so reconstruction of gene genealogies across the genome. You see evidence of long diverged branches that are suggestive of deep population structure, perhaps archaic admixture, and also other methods such as based on the conditional site frequency spectrum. This kind of U shape is only expected when we have population structure with introgression in the past. So it was at this time that Simon and I started working together with Brenna and Tim. We're here to think about. They were already thinking about exactly this question: is if we have. It kind of harkens back a little bit to early debates from the earlier in the 80s, 90s about trying to understand population structure among worldwide populations. But now we have this signal of population structure within Africa and many plausible models for what this population structure might look like. So this was a review paper that they published asking, would we be able to propose models that capture kind of this diversity of plausible models of history, parameterize them, and actually learn them from data. So comparing an African multi-regional model to a recent expansion model to an archaic atmosphere. So very briefly, we set out to test these things. We used some recently generated data from that we tried to include more genetically and geographically diverse populations across Africa. To answer this question, we read PCA and admixture like a lot of genetics papers do. But just to point out that I think this is also useful to demonstrate that these first 5 cohorts are the 1,000 genomes populations, and there's a lot more genetic diversity within Africa that is not captured in the 1,000 genomes data. So we were trying to take advantage of more genetically diverse populations and build models. So fast forwarding some years, we landed on models that look like this. So fitting parameterized models to the data across these populations, we find multiple plausible models. So different models, you could parameterize them in so many different ways. Model space is gigantic. But we think we found some common features that you can kind of tease apart from multiple models that fit the data well. One of them is that, yes, we do find evidence for ancient structure, but those past populations were likely exchanging genes, at least periodically. We found models that included migration between ancestral populations fit much better than models where we had isolation and then recontact. So this is something that maybe is a little bit different from other models that are being fit to data in the last few years. We also found that even though we have deep population structure, shared common ancestry doesn't extend much beyond, the deepest divergences among populations that we looked at doesn't extend to 300, 400,000 years in the past, but is within the last 100 or 150,000 years. And so we can take these models and also ask, do our predictions fit other features of the data, kind of in a somewhat independent validation? And yeah, we fit the CSFS very well, which other groups have used to fit models of ghost introgression. And we also do well at recapitulating things like the instantaneous coalescence rate or cross coalescence rates between populations. So yeah, so this was work that we were excited about to finish, but something that wasn't included in this, which I think could potentially add a lot of be very beneficial to these types of analyses, is the incorporation of ancient data. So all of the models that we inferred and methods that we developed rely on fairly large sample sizes, and we were using present day populations. It would be nice to be able to incorporate information from lineages that are represented by just a single sample. Now it's very challenging to get estimates of LD from single samples that are unphased, but this would be the goal. So there are already ways that people have tried to approach this problem of learning population history from single ancient genomes. You can combine them and compute F statistics across sets of samples and build what are called admixture graphs. These can get very complex, but they try to measure things like amount of drift along branches and contributions from different lineages in a say proportional sense. Or with a single genome, you could do something like run PSMC. But with unphased genomes, it's hard to know how to combine them in a MSMC on multiple sequentially Markovian process model. So this is work that's been led by Nick in my lab. He's a postbac researcher. He's been thinking really hard about how to, well, exactly this problem, how to get some extra information from haplotype structure when you only have single unphased genomes. And so what he realized, or what we proposed, is that you could take a fairly simple statistic, looks like this, we're calling it H2. It's essentially just taking two loci and asking, is your individual heterozygous at both sites? And you can be heterozygous if both of the derived alleles fall on one haplotype and you have ancestral alleles on the other, or the reverse. So you can have a coupling LD or the repulsion LD. And then at first we called this D plus, because it's essentially D, but with a plus sign. But we decided to spare you another statistic that's named D. But this has a really nice genealogical interpretation. See, it's still called D plus there. But under steady state equilibrium model, you would expect this to decay to theta squared at the when recombination gets very large, and 2 theta squared when recombination gets small. And the interpretation here is that this is capturing the covariance of coalescence times at the 2 loci. And so if you have very, very large R, very large recombination rate, you have effectively independent coalescences of the 2 loci, and your H2 just becomes the product of pi at both loci. When they're linked, when they're fully linked, they coalesce at the same time. And this leads to this extra covariance term. And so the covariance when they're fully linked is 1, 2 things squared. And it decays around this point of rho of 1, like a lot of these scale statistics do. This is sensitive to size changes, so you can model population expansions, bottlenecks. It has a characteristic shapes that are depend on the time since these population occurrences or demographic occurrences. And you can also extend it to cross-population measures of the statistic. Since we don't have phasing, you have to kind of account for all possible phasings, but you could build this into the model. It still gets signal. We show that actually this is really useful because you get not just single locus diversity, but something about haplotype structure in this 2 haplotype setting. So one exercise you could do is model something that has, say, a split with migration and recontext. So something like structure, you can come up with a single population model that fits that gives you the same instantaneous coalescent rate at single locus. And when you look at this decay curve, it fits at this. So we think that we have some information, some additional information in this statistic. It's also, however, a statistic that is sensitive to certain things like variation in recombination and mutation rate. So we have to account for those things, but we go through a lot of work to show that we can account for variation, local variation in mutation rates, local variation in recombination rates to use the statistic in an inference framework. So we set out to test this kind of as a proof of principle. We were interested in a set of questions that other people have looked at in the human, Neanderthal, Denisovan, ancient DNA world. So we were interested in this question is, do we see evidence for recurrent or bidirectional gene flow between humans and Neanderthals? Do we see evidence for a superarchaic introgression into the Denisovan lineage that people are thinking might be there? And we also asked about something a bit more recent, the formation of lineages leading to early Europeans. So we used at the time, the 3 published Neanderthals, the Denisovan genome, and 3 high coverage ancient Eurasians from Central and West Eurasia. So very quickly, I'm just going to go through these briefly. We find this is a question that has been asked in using many different methods. So work from Adam Siepel's group using ARG reconstruction, work from Sarah Tishkoff's group and Josh Akey's group. They see evidence for early human, so not just the Neanderthal to Eurasian gene flow event, but also early human to Neanderthal introgression events. They find different introgression proportions, so as low as 2% early on from Adam Siepel's group up to maybe 5 or 10%. So there's a lot of variation in the amount of this introgression. So when we fit these models, we see that we do find this signal as well. I forgot to mention that we also fit this into kind of this nice, Nick figured out this really nice framework for assessing statistical significance in a likelihood ratio test that involves a bunch of bootstrapping the data and refitting, but we find really strong support for both an early and a somewhat later series of introgression events. Importantly, we found that you do need to include migration between the Neanderthal and Denisovan lineages to get a good fit to those data. And also maybe this is on the higher end of the proportions of early human to Neanderthal gene flow. When we also looked at the super archaic introgression into Denisovans, so this is this potentially unknown hominin contributing something between 0 and 10% admixture into the Denisovan lineage. We also find evidence for this. It was supported. It was statistically significant. We get estimates greater than 0, but kind of at the lower end of about 1%. So that's this here. And notably, when we also include this feature, we get lower estimates of this human to Neanderthal, slightly lower, more in line with what Sarah Tishkoff and the lower end of Josh's estimates of around 5%. Yeah, and then finally, there's been a lot of interest in this so-called basal Eurasian lineage leading to the formation of Europeans. And so we parameterized a bunch of different models for how to think about what leads to these different European lineages, which are early farmers or Western hunter-gatherers, or these early North Eurasian groups that's represented by the Ustashin. And we do find also support for this in the data. And the admixture proportions are very similar to the inferred admixture proportions from the admixture graphs that were published almost over 10 years ago now. But we have from this method, we can actually build a demographic model that includes population sizes, actually date split times and introgression. And so putting this all together, this is a small statistic. You only use a single sample from each lineage. So that means that you can include a lot of samples. And so you can include all of these features and build actually a demographic model that incorporates these features that we think fits the data quite well from these statistics that we've looked at here. Okay, so some conclusions. We think that we can take these single representative genomes to infer parameterized demographic models, not just admixture graphs. So you can take these models and plug them into a simulation framework, plug them into like SLIM and actually simulate something like selection through these if you were interested in that. So it's potentially provides a tool for doing that. You can assess feature significance, the significance of parameters in a formal likelihood ratio test. Two points of caution whenever you in this method or really any method, these admixture proportions and timings get really confounded. We found very different values when we include, say, the super archaic introgression versus if we don't. And so to me, that tells me that, you can add features that you think are there and get better estimates of these values. But we also don't know what features were there that we're not including here. So I would interpret all these values with caution, because we don't know what other features are actually lurking in our history that would confound parameters that we think we're learning. And so that is to say that model space is huge when you include a bunch of populations. So it's the biggest source of uncertainty that we have to contend with. Okay. That was supposed to be like halfway, so I don't think so. I'll briefly talk about a little bit about selection, thinking about selection on introgress variations. So once you have one of these models of history, how can you think about what alleles were being introduced to populations through introgression events? Were they selected for? How strongly? Why? In some cases, we think there's been maybe positive selection. So this would be something like, it's called adaptive introgression, where maybe a beneficial allele was introduced, say to humans from Denisovans or Neanderthals, and then later selected for in some environmental setting. But we really think that most selection, if it occurs on introgress variations, is probably negative, deleterious. So one of the arguments might go something like this, where you have, in a simplified model like this, you have a large population, a small population. In that small population, you have slightly deleterious variation that is allowed to accumulate over many generations. So you would have these slightly deleterious alleles accumulating, and then when they're reintroduced to a larger population, they're exposed to selection and selected against, and then thus removed in the recipient population. And so one potential point of evidence for this is regions of the genome that are largely devoid of introgressed ancestry, so-called introgress and introgression deserts. So those are regions that we don't see Neanderthal or Denisovan ancestry. The mechanism of selection is, if it actually is selection, is yet to be determined. But we also see evidence in the depletion of Neanderthal ancestry in regions that are highly gene dense or in putatively regulatory regions. And so we see a depletion in functional regions of introgressed ancestry. There are other possible mechanisms that can lead to selection against introgressed ancestry. One of those is genetic incompatibilities, where you can have the say incompatibility loci that get fixed for alternate alleles that aren't tested against each other when they come back into the same population, and you would only have selection once they see each other within a diploid individual. And the way these get resolved is selection against the minor ancestry. So you get the removal of the deleterious or the incompatibility allele that accumulated in the Neanderthal branch. So what happens in these settings when you have reciprocal gene flow? We now think we have evidence for it. What about human alleles being introduced to Neanderthals? Well, the load argument would say that, well, the human haplotypes are more devoid of deleterious mutations because they've been maintained, those haplotypes were maintained in a larger population for longer. And so they would be favored in the Neanderthal back and increase in frequency. And so there's maybe evidence of this on the Y chromosome and the mitochondria, which look like they were potentially replaced. The Neanderthal haplotypes in the uniparental chromosomes were replaced by the human haplotypes after introgression. And that's the evidence for that is you see, like you see lower coalescence times of human and Neanderthal mitochondria. They coalesce first before they coalesce with the Denisovan, and more and young and at a younger age than the population split of the common ancestors of Neanderthals and Denisovans. But other models will make different predictions. So the incompatibility model, well, if you introduce this incompatibility allele into the Neanderthal background, if this is a low introgression proportion, you have selection. The way to resolve this is selection against the minor allele. So you would actually get selection against the human haplotype at incompatibility setting, and so forth. And some recent work from Sarah Tishkoff's group set out to test this. They think they were able to, or they tried to call human haplotypes in a Neanderthal individual along the autosome, and then asked, do we see an excess of human alleles in these functional regions or a depletion? So do we see kind of one of these cases or the other? We see more human haplotypes where we have introgression deserts and movements or vice versa. And they find evidence for that the human haplotypes in the Neanderthal background are depleted in these same regions where the Neanderthal haplotypes are depleted in humans. So they think this is evidence for an incompatibility selection model rather than low or unconditionally deleterious alleles. But I'm of the opinion that this is a fairly short amount of time for incompatibilities to arise and fix in these two lineages. So what is leading to this signal of incompatibilities? And something that a lot of these models don't include are models of, well, just what we think of kind of the natural process of kind of the everyday process of selection on quantitative traits, which is, we think that a lot of traits are under the action of stabilizing selection rather than directional selection. We think that gene expression is often subject to stabilizing selection. A lot of different traits are subject to stabilizing selection. And so it's natural to ask, how does stabilizing selection impact these dynamics of selection after admixture? So stabilizing selection is a process where individuals who are, there's some phenotypic optimum, and individuals who fall farther away from that optimum have reduced fitness. And so you have selection maintaining phenotypic distribution of the population around that optimum. And it's going to depend on the strength of stabilizing selection and other features of the genetic architecture. For the sake of time, one thing we know about stabilizing selection is that the turnover of alleles, the turnover of the genetic architecture for alleles that contribute to stabilizing selection is faster than under this drift alone. So yeah, we think that alleles are going to arise and perhaps go to fixation or loss faster than just under drift. They also have a higher chance of fixation than an unconditionally deleterious allele. So it's stable. It's selection on alleles contributing to these traits is not as uniformly deleterious than just directional selection. So they have a higher chance of fixing in the two lineages. So we expect that there's going to be this rapid turnover and potentially in this time some amount of fixed differences or at least allele frequency differentiation at these at the trait affecting loci. These two lineages. So yeah, so I'm just going to very briefly point out, I'm going to skip like 30 slides. And so we can ask, do we see this effect, say, so I have a bunch of model-based approaches where we can predict allele frequency changes in a site frequency spectrum trait based background. I can also predict the dip in allele frequencies around trait affecting loci due to linkage. And so we can ask in a model like this, where we have functional regions dispersed along the genome with bidirectional gene flow. Do we see evidence of this, at least in a modeling framework or a simulation framework? And we do see this. We see if you simulate stabilizing selection in a multi-population framework with somewhat plausibly human-like parameters, human Neanderthal-like parameters, we see depletions in ancestry in and around these functional regions, both in the Neanderthal background as well as in the human background. And if you ask, where are you more likely to see introgression deserts, very loosely defined, you see that they're more likely to be in those functional regions, and they're also more likely to be shared across the two backgrounds. Yeah, so this is opposite of the prediction that we would make under load. I think it's also opposite of the prediction that you would make under like your classic Dubjansky-Moeller incompatibility model, where these are resolved via selection at different, where this is a way that you can get selection against those trait affecting ones of the same. Okay, so I'm gonna wrap up there. I'd love to talk a lot more about stabilizing selection and introgression. Talk about stabilizing selection. Also would love to talk about stabilizing selection when you have differences in population sizes and what that means for fitness and load and these types of questions within populations, and how that kind of, I think, reshapes how we think about selection operating in Neanderthals. But for now, I'm gonna stop there. Take questions.
Yeah, just a clarification on the last part. In humans and Neanderthals, is it still stabilizing around the same optimum?
Yes, yeah, so everything is consistent, same optimum, same mutation rate, effect sizes, everything, just so it's different as the demographic processes.
Cool talk. Did you see a poster at ProbGen about ghost introgression that was, first of all, does that ring a bell?
Yeah.
Okay, and they had, yeah, okay, sorry for, if I remember right, they had called tracks of ghost introgression, and they made a point that they were in the Denisovan and Neanderthal deserts. Yeah. And I was wondering how you thought that would fit into your mind, because that, right, depending on the shape of the tree and the amount of time that you would have for things to evolve, that doesn't really track with, yeah, that again, like informs whether we're talking about universally deleterious versus DMI, or, and then this model, I guess it depends on the shape of the tree and the divergence time. But yeah, if you had seen that and already thought about it, I was wondering if you had any thoughts about that particular idea and how it relates to your ideas.
Yeah, so the signal is, if you look in these regions where you have Neanderthal and Denisovan deserts in humans, when you look at where these are from a ghost admixture in African populations, you see that these are interspersed across these regions. Yeah. I think that there are a couple plausible, well, part of the problem is that we don't know what actually forms these introgression deserts from Neanderthals and Denisovans. It could be just stochasticity due to bottleneck effects if these were small populations and these were stochastic regions of stochastic loss.
Yeah.
There are selection models that could explain it. I don't think you can discount that there were inversions in the Neanderthal or Denisovan lineage that lead to this. That wouldn't be shared with potentially this ghost ghost model. So there are a lot of possible models that could.
Sure, yeah, I know there was a lot going on there, but I thought of that watching your talk. I was curious if you had seen it.
I think it's an interesting data point to try to fit into these models and might constrain what we think are more likely models. But since Friday, I haven't had a
sure. I mean, it's not that long.
Yeah.
I think I'm still wrapping my brain around all of it, so this might be a silly question, but if you're seeing, so the signal of stabilizing selection is also, partially defined by this more rapid turnover, but selection around the same optimum. Sounds like a scenario that would lead to more incompatibilities hypothetically, because you're, replacing alleles with new alleles and different alleles in the two populations, and they're with each other, but then when you get into regression. So I guess you were like, we would see opposite patterns, and my brain does not immediately compute how that's possible, because it seems like one could cause the other.
Yeah, I guess it's a little bit semantic if you want to think of like what this is in this model, what this is doing with stabilizing selection is in individuals that have ancestry from both lineages, you have higher genetic variance. And so selection is increased for some amount of time to try to bring that distribution of phenotypes back towards the optimum and squeeze it back in, and in doing so, it removes the alleles that are kind of pushing you away from the optimum, which are those introgressive in the recipient genetic background. I don't think those aren't like maybe classic DMIs, like you're looking for mitonuclear incompatibilities or these sorts of things, but it's kind of weak epistatic interactions that are caused by that stabilizing selection causes anyway, that are slightly more exacerbated in the hybrid individual. So I don't know if that answers your question.
Yeah, I think it does. Yeah, it helps. Trying many questions.
I'm thinking a little bit about connecting the last two parts of your talk, and I'm wondering how reliable these putative human integration deserts are in the archaic genomes, given that you've only got about 3 or 4 high quality genomes. And then, in the middle half of your talk, you mentioned how you do infer gene flow from, an ancient African population. But I mean, depending on exactly when that was, how long this, but they're going to be very short haplotypes now. They're going to be hard to infer. The archaic genes are gappy. So yeah, I'm just thinking a little bit like, do you really think that these are truly human deserts in the archaic genomes, or you think this is more of a
what the signal is a statistically significant depletion. Like this is 5% of the Altai genome. If you believe the introgression proportions, roughly 5% will be human haplotypes in the Neanderthal. And so I think what they do is they do kind of something like a permutation test, where they're asking if you were to randomly place these, given the recombination map and et cetera, do you see is the distribution within the Neanderthal pattern?
it's like a haplotype matching that they're doing. But it's a haplotype matching they're doing when gene flows occurred 300,000 years ago.
Yeah, they base it on, they have to, there are a lot of assumptions. So they're using some haplotype calling algorithm that uses a reference, and this reference is a set of sub-Saharan African individuals and their genomes that they think don't have negligible Neanderthal ancestry. And then these are called with some support.
Yeah.
I think there's a lot of I think you can look at this and wonder how reliable those calls.
I'm just disappointed in the loader.
I think, well, what I think and what I didn't get to is both of these processes are probably at play, and so just trying to think the trick is going to be how do we fit these models together, because we think we know stabilizing selection is acting on a lot of traits. There are unconditionally deleterious mutations that occur. So how do you fit these together? The story for the Y and the mitochondrium is probably different than the autosomes. Probably the X is different than the autosomes. So fitting these together is, I think, an interesting direction to go. Yeah.
I was curious on the demographic inference bit. You mentioned how large the model space was, and it seems like there are a lot of choices one has to make. And I imagine the inference bit is not computationally trivial by any means. I was curious, like, to what extent can you, one, is it feasible to test like sensitivity of these things to at least say like you mentioned, like, you know, qualitatively these things match to other types of methods, like, is sensitivity analyses of these things like feasible under this model space? And if so, like, what does it kind of look like? Does it look like they?
Yeah, so what we did is we explored a very narrow area of parameter space where we're specifically asking if you have a model of divergence, you can your null model might be something without introgression, and then you're testing for support for that introgression. You can think of so many different topologies that connect these populations and migrations, whether they're there or not, that it gets kind of impossible to explore it all. One thing that is useful to do whenever you infer a demographic model using one inference framework is asking you, are you able to predict statistics that are used by a different framework or different somewhat orthogonal features of the data? So we tried to do that in the earlier paper looking at population structure of the stems of humans, seeing if we can predict other features that we didn't use in that demographic inference. And if you do predict those well, then that's at least suggesting you're on something like the right track. Yeah. But it's not maybe not a formal sensitivity analysis.
Yeah, I was wondering the same thing. Yeah, like it seemed awful to try to get all the relevant topologies. So yeah.
Yeah, a lot of the time is spent trying to think what are features that you're not including that are plausible that you should test. Try to explore this. Yeah.
Yeah, in a similar vein, well, first of all, I really appreciate your being so honest about the problem of model misspecification, and so thanks for doing it. But I still want to hear what you have to say about how you get a sense of when you're kind of close enough. When are you close enough to something that's a really good model, and how would you compete a couple of different models? And I think you answered the question just then, a narrow question about the likelihood ratio test. So it sounds like your test is a test of, is the feature present or is it absent? Mm-hmm. So that it's encouraging that you even have a likelihood sitting around. That means you've got a loss function, and so you can get started. What would you like to be able to do in this regard of competing models and examining goodness of fit and complexity? What would you like to be able to do that seems attainable, that you're not quite able to do yet? And is there any chance of getting something like a Bayes factor in here?
Good questions. I don't know. Possibly. I don't know about that.
It's always hard to get a Bayes factor, and it's starting to become more common.
Yeah, that would be. I don't know is the answer, but
yeah.
To your question about what would
you like to be able to do that you're not that seems attainable, but you're not quite there.
Yeah, these models are very boutique in the way that we're like they're tailored to the question you're asking, and like the models that we proposed were based on models that have been proposed by other people in the literature. It would be really nice to have a way of more a more automated, and I think this is doable, a more automated way of testing different topologies. Maybe your learning parameters conditioned on the models the topology in the model that you propose, but a way of maybe sampling different topologies within this framework and without having to do it manually every time based on what you think is possible. Because I think this is done more often in maybe a phylogenetic inference framework where you detach edges and reattach them and test different model topologies. Having something similar here is think doable is just maybe computationally challenging, but not conceptually challenging.
Maybe we'll end there with the promotion of artisanal population genetic models. And thank Aaron one more time.